Okay, so in this video, let's zoom in on these neurotransmitter receptors a little bit. Before going into the structure, let me give you some general properties of neurotransmitter receptors, just that you can just memorize, or you can try to make sense of mechanistically later on, which is the way I would prefer to do it. But we can think of a couple of dichotomies with respect to those receptors, very clear-cut categories. The so-called fast and slow receptors. And fast is just an ion channel. So neurotransmitter gated ion channels, once the channel is active, ions flow through in immediately. As opposed to the slow channels, which, as I mentioned earlier, are G protein coupled receptors. So their response is ultimately going to be some change in potential, but it's going to be mediated through the G protein, probably the beta gamma, activating some channels. So that's going to be slow, no matter how you look at it. And then each of these categories can be subdivided in terms of the outcome of the activation, so that you can have a fast depolarizing event, say opening a sodium channel, and that would be considered excitatory. Or you could have a hyperpolarization event in which the potential becomes more negative than the resting potential, potentially, and that would be considered inhibitory. And again, you can just memorize these connections between excitatory and depolarization, inhibitory and hyperpolarization, or I'll just try to explain it mechanistically, so that will help you memorize things. And exactly the same categories would fit here. And we can further identify the nature of these outcomes as far as active neurotransmitter receptors. Depolarization would be caused by the opening of a sodium channel, which would trigger the movement of a positive charge from outside to in down the sodium concentration gradient. That's pretty much the only kind of depolarization we're going to talk about. Inhibitory responses or hyperpolarizations could be achieved either by opening a potassium channel, in which case potassium would move out, so a positive charge moving out across the potassium concentration gradient, which would be a hyperpolarization, just like you have with the action potential. Or you could open a chloride channel, in which case chloride would move in, but because chloride is negatively charged, that would lead to the overall contribution of making the potential more negative, and that's considered a hyperpolarization, and that would inhibit in a way that I'll get into in just a minute. So in terms of the G protein coupled receptors, exactly the same things apply. Excitatory is opening sodium channels. Inhibitory, which is the more common event, is the opening of a potassium channel more often than not by the beta gammas. Okay, so let's move over here to the specifics of the fast channel architecture. And I already alluded to this earlier. Here is the acetylcholine receptor. In this case, acetylcholine is a neurotransmitter, and this is a fast receptor. It is a fast excitatory one, which would lead you to guess correctly that it is a sodium channel, that when it's open, sodium goes from outside to in. And the reason it's a sodium channel and not some other ion can be gleaned from looking at the nature of the amino acid residues that line the pore on either side. Because if we're talking about a cation channel, then those are going to be negatively charged things, and something like a chloride channel, those would be positively charged, so that would be a giveaway. The rest is just geometry and Eisenman when it comes to determining the specificity of this particular event. So here's the gate that is going to be controlled by the neurotransmitter itself. So this is the acetylcholine receptor, the so-called nicotinic acetylcholine receptor, When it binds, it triggers the movement of residues in that gate region so that sodium can go flooding into the cytosol and trigger the excitatory depolarization. So why is a depolarization excitatory? That's something that is shown not from a diagram in your text, but from another diagram that I think has this described pretty nicely. So again, here's our cartoon of a neural circuit. So this would be the pre and postsynaptic cells where we've been. So the same area would be shown here. Action potential moves along the axon terminus, arriving here along the axon to the axon terminus. Acetylcholine, let's say, is released, or glutamate, something like that, and then bound by neurotransmitter receptor here. So those events, excitatory or inhibitory, are going to be determined by not one, but in some cases hundreds of thousands of presynaptic cells that are weighing in, releasing different neurotransmitters. Typically a single neuron is going to be associated with a single type of neurotransmitter, but there may be thousands of different types of neurons associated with this particular cell, and they can commonly do send different signals. So it's the job of this neuron to assimilate all that information, and what it does is basically assimilate that through a series of excitatory or inhibitory events. So if this is an excitatory neurotransmitter receptor and neurotransmitter at this synapse, then you'll get a depolarization, and that's going to contribute to changes in the potential, mostly that accumulate here at the so-called axon hillock of the postsynaptic cell, that or as depolarizations are, makes the potential more positive than the resting potential. So let's say that you have a series of excitatory neurotransmitter molecules released by the presynaptic cell or cells. That would lead to a series of depolarizations. Once that potential at the axon hillock has reached a certain threshold, and that's going to differ with different neurons, but let's say it's minus 40 millivolts, then you get the firing of those voltage-gated sodium and potassium channels, and the action potential moves along this axon. So if, on the other hand, the neurotransmitter or neurotransmitter in question, receptor in question, are inhibitory, then you would see the potential drop more negative as a result of that event. So you might have a series of excitatory events followed by inhibitory events that drop the potential back to close to the resting potential, in which case this neuron is not going to fire and might even be prevented from firing by inhibitory events. So this is the sort of thing that we would associate with active potassium channel neurotransmitter receptors, or more commonly chloride channel neurotransmitter receptors. So things like the GABA receptor, that is very important pharmacologically. Something like Xanax would stimulate that receptor, but by stimulating an inhibitory receptor, what you're doing is you're calming the neural circuit. You're dealing with anxiety caused by an overactive stimulation of a nerve circuit through the addition of something that stimulates an inhibitory neurotransmitter receptor like the GABA receptor. So that overall effect would then modulate the amount of electrical and chemical communication that is going left to right in this circuit. So what we have yet to accomplish is making this muscle contract. I'm not going to talk about that much because we're pretty much out of time, but I do want to talk a little bit about what's different about this particular synapse than this synapse. And while I'm on this level, we can talk about some terminology that would distinguish those two synapses. So what's happening in the case of the communication between the interneuron and, let's say, a motor neuron, is that an excitatory event is communicated from the pre to the postsynaptic cell. So in that sense, we refer to this type of coupling across that synapse as EE coupling. I think there's a dash there. So that would be that would stand for excitation coupling. And that's a description of the mechanisms that go into describing this particular synapse. At this synapse, what's happening is electrical communication is converted into the contraction of this muscle. That's done through depolarizations, but what's ultimately the outcome is contraction. So we refer to that synapse as participating in EC coupling, excitation-contraction coupling. And that involves a very complicated series of lectures that would describe how muscles contract, and I'm just not going to have time to do that. I'm hoping you learned that in some other physiology class, like human physiology, and we'll just focus on the cellular mechanisms and be done with it. And those are shown in this diagram from your text. So what's happening is the synapse that links the motor neuron to the muscle is shown here. So here's the motor neuron. An action potential of a very standard sort arrives at the axon terminus. Voltage-gated calcium channel is waiting for it the way it always is. Acetylcholine, and the typical motor neuron, is waiting in tethered and docked but not fused neurotransmitter vesicles. So when the action potential arrives, calcium comes in as it would through a voltage-gated calcium channel, because there's more calcium here than here. That triggers essentially the approval of fusion here, releasing acetylcholine into the synaptic cleft, where it goes across to the postsynaptic cell and encounters that acetylcholine receptor, a fast receptor that when it's open, it allows sodium in. So that's the structure that we saw in a previous panel. So when sodium comes in, it leads to a depolarization, making the resting potential deflect toward a more positive number, very reminiscent of kicking off an action potential. So that's shown here in terms of the opening of a voltage-gated sodium channel as a result of that depolarization. But muscle, at least skeletal muscle, has these infolding of the plasma membrane in the form of so-called T-tubules that I hope you've learned at anatomy or physiology. I'm not going to talk about what they are, but they are invaginations of the plasma membrane. The point being that you create a particular structure that links the plasma membrane with the very specialized endoplasmic reticulum of skeletal muscle, which gets its own name, the sarcoplasmic reticulum. And like the ER, calcium is sequestered in here. So we have calcium here in the off state. And we have a particular arrangement of proteins that one is an integral membrane protein of the plasma membrane, one is an integral membrane protein of the sarcoplasmic reticulum. Your book calls this a voltage-gated calcium channel. It looks like one, but it is not a calcium channel itself. It is actually the dihydropyridine receptor, the DHPR, and it is physically attached to a calcium channel in the sarcoplasmic reticulum that your book calls the calcium release channel. Most people call it the ryanodine receptor. So in the off state, calcium is here and here. When a depolarization comes, it goes along the surface, dips into the cytoplasm through these T-tubules, and activates DHPR, which then pulls open the ryanodine receptor that makes the calcium go out from the sarcoplasmic reticulum into the cytoplasm of the muscle cell. So that sequence of events is shown in the numbered sequence here. Calcium goes in, triggers the release of the neurotransmitter, binds to the neurotransmitter receptor. Sodium moves into the muscle cell, triggers a depolarization that is responded to by the dihydropyridine receptor, leading to the opening of sarcoplasmic reticulum calcium channels. That dumps calcium onto the cytoplasm, which, if you know your muscle anatomy, we'll make it red for muscle, there are myofibrils here, which means that there are those alternating actin and myosin filaments. Basically, what happens is the calcium, when it's dumped on those myofibrils, it triggers the contraction by a series of conformational changes that allow myosin and actin to interact with each other, and the myosin power strokes. I'm not going to talk about those at all, but the bottom line is the calcium release shown here is going to trigger contraction here, essentially allowing the myosin power stroke to do work by contacting actin. And that is the last step in this overall series that began, remember, with slapping the sensory receptor. So we have the accumulation of probably depolarizations as a result of that event, leading to an action potential moving along here, triggering the release of a neurotransmitter here that is recognized by the dendritic cells of the interneuron. Eventually a threshold is reached here at the axon hillock, sending an action potential along here. Neurotransmitters are released, recognized by the motor neuron. The axon hillock counts, and then once the threshold depolarization occurs, fires in the form of a series of active voltage-gated sodium channels followed by potassium channels, blah blah blah. Calcium channels open, acetylcholine is released, ultimately triggering muscle contraction here. And that's that.